Hey everyone, welcome. I'm going to finish up our macromolecules chapter 2 lecture with this video. So I'm just getting those slides ready, pulling those up. Hopefully you're pretty familiar with the 2 to 2.4 material by now, and are feeling okay with that. You watch that lecture first so that we can jump into our macromolecules. So let's see. I want to start. We finished last time with the pH scale and talking about, and I'm just summarizing this real quick, because this connects to our next stuff that we're getting into, the macromolecules. So we talked about how pH is a measurement of how many hydrogen ions are in a solution. Hydrogen being that H plus. Sometimes we call them protons, sometimes we call them hydrogen ions. It's the same thing though, right? It's an ion because it has that positive charge. And so we said that the pH is, that acid base, this acid base scale we're seeing. And as you have more hydrogens, it's more acidic, it's a lower pH. As you have fewer hydrogens, it's less acidic, more basic, higher pH. So where that comes into play is with hydrogen bonds, because it's interesting to see the pH of things like we see on this scale, but what does that actually mean in practicality in the body physiologically? Remember, blood pH is about 7.35 to 7.45, and we have buffers to really regulate that very tightly. Because if you start to adjust out of that, you start to fluctuate the number of hydrogen ions that are in the solution. If you're getting too acidic, if you go below the 7.35, you have excess number of hydrogens. If you get too basic, you don't have enough hydrogens, and that starts to impact hydrogen bonds. And if you remember hydrogen bonds, go back and review those sections. But hydrogen bonds play a huge role in macromolecular structure. If you remember something like methane, methane is a polar molecule. It's carbon with, it's a non-polar molecule. We have carbon with these evenly spaced hydrogen ions, all very balanced, all very even electronegativities. And so you have this very kind of straightforward molecule. Organic molecules, macromolecules, are much more complex. They have much larger structures, and because of that, they have much more complex functions. Remember, that always goes together. Water is a very important molecule, but it's a very simple molecule, H2O, just a very simple molecule, and its functions are pretty simple. It sticks to itself, it sticks to other things, it's pretty stubborn, it doesn't like being heated up or cooled down, right? But when we think about something like a protein or DNA, these large macromolecules with really complex structures, they have very complex functions. And so those things go together. And one of the big structural things that holds macromolecules together are hydrogen bonds. And we'll see, especially with proteins, as we start to adjust pH, if your body, let's say, is not able to balance your pH and it starts to shift too low or too high, your proteins start to break down and you die. That's what happens. So here we see 3 of the basic macromolecules. There are 4 different types of macromolecules. You have your carbs, proteins, lipids, and nucleic acids. Here you can see 3 of these, and we're pointing out that these 3 are monomers. And that's an important term that we're going to have to remember, is that a mono means one. And so a monomer is one single unit. And with macromolecules, we polymerize. We take one monomer and we add them all together to make a polymer. And so that process is called polymerization. And so you can see here with carbohydrates, you have one little carbohydrate monomer, a monosaccharide, that's been joined into a chain, and so making this polysaccharide. Same thing with proteins. Here's one amino acid. We string those together. With DNA, RNA, we have a nucleotide monomer that we string together. And so you have these small individual pieces that can be made together to make larger pieces. And they're going to vary. Each of these little monomers can vary, either be the same thing as its neighbor or it could be a different one. And that's going to give the macromolecule more specified functions. So we see macromolecules in our foods. Sorry, my slides are not clicking. There we go. We see macromolecules in our foods, carbs, proteins, fats, very common. You hear that a lot if you're, it doesn't even matter if you're into diet culture or you are around food science or nutritionists. If you hop on social media, you're going to hear a lot of information about food science, and a lot of it's going to be wrong. I could easily get on a soapbox and do a whole semester about that kind of stuff, but I'm not going to. That's not what this class is. But we are going to get into some of that because it's a science class. So you're going to learn some basic science, and hopefully that allows you to be a little more critical with some of the videos that we see on our feeds. So organic molecules are in foods, like we said. Some can be considered healthy, others associated with other conditions and diseases. I don't like making foods good or bad. I think that starts to provide big stigmas because it's important you get enough calories. That's really what it comes down to. Is you definitely have to have enough calories. If we can get good nutrition, that's a bonus for sure. And so there are foods that our bodies will react to differently because they have physiological components, right? The reasons that we say some foods can need to make us feel better or feel worse, it's because of how they're interacting with our bodies. Once our bodies absorbs them and it breaks it back down into those monomers, those then start to get incorporated into our cells and we start to feel different because of that. Okay, so I'm jumping to this table. So this table is really important for you to review. It lists the different types of macromolecules, their different subcategories, their chemical structures, and their functions in organisms. So make sure that you are familiar with this table. Do whatever you need to do to be able to rewrite it or have people quiz it on you or however you like to do that. Redraw it into a flow map if you want to, into like a bubble map diagram. That's a really nice way to kind of take something like this that's linear and maybe make it a little more interactive for yourself. So with macromolecules, I think it's important. I like to use the analogy of baking. I love to bake. One of my favorite things, I love making cookies. Cookies are really fun. They're very easy, very accessible, really quick recipes. So if you're trying to get into baking, start with cookies. Four basic things: flour, sugar, butter, and eggs. You got those four things, you can make a sugar cookie, you can make a basic cookie. And that's usually the base recipe for all cookies, for most cookies, right? Is flour, butter, sugar, and eggs. Well, there's multiple types of sugar. I can use just regular white granulated sugar. I could use powdered sugar. I could use brown, like dark brown sugar, light brown sugar, right? There's all these different kinds of sugars. Same thing with flour: bread flour, cake flour, all-purpose flour. With eggs, you have both the egg yolk and the egg white. So you could use maybe whole eggs and egg yolks for a little more protein and flavor. Egg whites have a lot more water content in them, so they tend to make things spread. With butter, you can use like room temperature butter that's actually still in the block. That helps your cookies to stay together more. If you start with liquid melted butter, which a lot of recipes tend to have melted butter, your cookies will spread more. Even if you chill the cookies, the dough is going to spread more as you bake because you started with melted butter. So it's really, really fun science, really, really easy to see applicable science in that. And so if you are interested in doing a cookie science lab, maybe that could be a fun one we can try to put together. But I want you to think about that kind of that analogy. That all right, when I can pick and choose different types of ingredients. I mean, I'm going to use sugar. What exact sugar ratio do I want to use? Do I want to use all white sugar? Do I want to use a mix of white and maybe some dark brown sugar, so that way I get that caramely flavor and that chew with also solving the crunch, right? Sorry, I could really get into cookie science. And so that's how the macromolecules are. We can have carbohydrates, but they can come in different categories of them. We have proteins. Proteins do so many things. All the work of cells are done by proteins. So proteins can be for transport, they can be for immune function, they can be for cleanup and digestion, right? So there's all these different kinds of subcategories within the one macromolecule category. So we're going to get into that. So again, hitting this idea that organic molecules, these macromolecules are made of monomers. We're going to look at the different, that's usually how we're going to start with each one of these macromolecules is we'll talk about its monomer form. We're going to get into those dehydration synthesis, hydrolysis reactions. So again, if you need to go back and review that, you can, because I'm not really going to review it. We're going to just go through it. And so we're going to see how we can put together monomers to make polymers. So here again is just that reminder that dehydration synthesis, listen to the term, you're removing water to put things together. We're joining monomers into polymers. Again, mono single, poly multiple. And so you can see here, we take these smaller individual monomer units and we join them into a polymer. This is an anabolic reaction. We are building things up. It is anabolism. We are putting energy into these things. We're storing energy into our polymers. And so this is a synthesis anabolic storing reaction. Hydrolysis is going to break things apart. So if you need to break things back down, like when we do digestion, when you eat, when you go to consume food, yeah, let's say you're eating chips, right? Chips are mostly just carbohydrates. Your body can't absorb that entire polymer. Your body throughout your GI tract is going to be breaking it down into smaller and smaller units until it gets down to that monomer form. And that's what our cells can absorb. If you eat chicken, right? If you eat chicken breast, you have a nice healthy chicken breast protein, your body is not going to absorb that entire protein. It's going to break the protein down into the amino acids and absorb individual amino acids. And so our body has to be able to hydrolyze, has to be able to break down down our polymers back into monomers. These are catabolic reactions, catabolic catastrophe, breaking things apart. These are releasing energy when they do that. So rather than storing energy like synthesis, we are releasing an energy with hydrolysis. So let's start with carbohydrates. Carbohydrates are very, familiar for most of us, I'm sure. Things like simple sugars and polysaccharides. Simple sugars are monosaccharides, disaccharides, things like table sugar, right? Very simple sweeteners that you're familiar with. Here are three monosaccharides: ribose, glucose, and fructose. You can join these together to make disaccharides. You put glucose and fructose together, you make sucrose, and that's a sweetener, right? That's pretty much what they are. We see ribose. Go ahead and think about where you're going to see ribose in just a little bit, if you're already familiar with our macros. So with the reactions for synthesizing carbohydrates is you take these two monomers, these two monosaccharides, again, a glucose and a fructose. You see this these two hydroxide groups. We're going to dehydrate it and put it into the sucrose molecule, and then we can hydrate it, go through hydrolysis, adding water. Like if you put sugar into water, right? You put sugar into water, you stir it around, it's going to separate out into its monosaccharide components. There are again, two main categories of carbohydrates. You have your simple, which are your monosaccharides, disaccharides, just kind of sugar and energy, pretty straightforward, right? You use glucose for energy. The complex carbohydrates, they are, again, more longer structures, right? These are your polysaccharides. These are ones that are fully polymerized. You can see things like cellulose, starch, and glycogen. You probably heard of two of these, probably haven't heard of glycogen very much. But notice that these are, again, still monomer chains, but they are much, much longer than just one or two monomers. These are polymerized chains, 20, 200 plus monosaccharides together. So while your simple sugars are used for for energy, right? We can easily break down those to use them for quick energy sources. You eat, some candy or some chocolate or some, again, go back to chips, I guess. I just had some chips before this. That's why they're in my brain. But just have some chips. Your body can easily break that down into quick energy sources for sugar. Polysaccharides, more complex structure, more complex functions, harder to break down. This first one that we're seeing is cellulose. Cellulose is found in plant structures, plants for structure. If you think about leaves and stems, trunks, right, the woody stick structures of plants and trees, that is all cellulose. Starch is the energy storage for plants. So you're showing a potato here. If you think about like apples and bananas, the part of it that you eat, if you think about like nuts and things like that, right? That's where the place where plants store their energy, store their food. Usually they have seeds with them, so that way those seeds can use that starch as an energy source, right? An apple falls from the tree, maybe something, it's sweet, so something's going to eat it, that'll help consume it. That apple has that energy around it, that glucose around it to be able to absorb, the seed does, to be able to help fuel itself to grow. And so plants will use both your cellulose and starch. Plants make cellulose and starch for cellulose for structure, and then starch for energy storage, right? We use proteins for our structure. Plants use cellulose a lot. The other one is glycogen. This is the other complex carbohydrate. So three complex carbohydrates, cellulose and starch, are found in plants. Cellulose for structure, starch for energy storage. Glycogen is found in animals for energy storage. So we'll use simple sugars, glucose, for quick energy if we need it. But if you are at rest when you're eating and then your body doesn't need to use that energy, that glucose immediately, your liver is going to convert glucose into glycogen so that it can be stored there and in your muscles for later use. Okay, so simple carbohydrates are monosaccharides, glucose, fructose, disaccharides, sucrose. Our body uses these for quick energy. Complex carbohydrates, polysaccharides, there are three classes: cellulose and starch in plants, glycogen in animals. Starch and glycogen are energy storage, cellulose is structure. Okay. So again, if the plant or the animal is producing a lot of glucose or is consuming a lot of glucose and it doesn't need that immediately, it's not trying to grow or reproduce or make anything, then plants and animals will take that excess simple sugar, polymerize it into these complex carbohydrates where it can be stored. Okay, so let's jump into proteins. Proteins are the most diverse macromolecule. They are the most numerous macromolecule in your body. I always say, if you aren't sure about a question and that question is asking you what macromolecule is responsible for this and you don't really know, guess a protein. It's probably a protein. They are responsible for most of all of the work in a cell, in organisms. So proteins, you can see some different classes here: structural, antibodies. Structural is in collagen, collagen's in your skin. Antibodies, obviously for immune protection. Contractile proteins for muscle, both in like skeletal muscle, contracting those. Blood vessels constricting and dilating. Transport membrane channel proteins, you have to get stuff in and out of cells. So proteins provide for movement across the cell, storage and albumins, and enzymes. Enzymes are proteins, they aid in digestion, they are necessary, required for life, not just digestion, but in everything we do. So proteins, again, most diverse of our macromolecules. The monomer form of a protein is an amino acid. So there are 20 different types of amino acids on Earth. There are 20 different amino acids, and all organisms use some combination of those 20. Humans, mammals, most complex animals use all 20. We need all 20 amino acids. Smaller organisms, some plants, some bacteria, they don't have all 20. They're going to have some of them. But humans, we need all 20 of them. And what organisms can do is we take these amino acids, these 20 different amino acids, we can put them together in different combinations. And because they have these R groups, these functional groups, they have different kind of qualities, different hydrophobic or hydrophilic, positive charge, negative charge, small or large molecules. That all impacts how they interact with each other. And we're going to see that interactions, intermolecular interactions, cause proteins to fold into a unique functional shape. Okay. So here is the general structure of amino acid. You have this central carbon atom, so there's always going to be a carbon right in the middle, and then you have two groups off to the side. This one over here with the carbon and oxygen is called a carboxyl group, so if you want to say that with me, a carboxyl group. And then over here on the other side is your amino group. Amino. And if you ever see this word amino, amine, whatever, any kind of derivative similar to this, usually that means that there's a nitrogen in there. And so your nitrogen is your amino. And I remember that because nitrogen, N-I-T-R-O-G-E-N, N-I-O, and amino. So there's three of those nitrogen letters. And so that's how my body remembers amino is nitrogen. It's simple, it's silly, but it works for me. So amino acids are always going to have this nitrogen-containing compound, a carboxyl group, right? Those are standard for all of them. But then this R, this is just a placeholder. I don't know why. They could have picked another, A group, B group, but they just chose the R group. This is what gives each amino acid its individual function. And so if you do like biochemistry, you'll have to learn all the different amino acid structures because you'll have to look at how they interact with each other. We're not going to get that complex. We just want you to kind of remember, all right, you have the amino group, the carbon, the carboxyl, and then this R group up there that's going to influence how amino acid works and interacts with its neighbors. Here's just some showings of that, right? So you see this simple hydrogen, this one with a sulfur is called a thiol group. So this is a thiol group, it's called cysteine. And then this one has this really, really large, these benzene rings up, this benzene ring up here, and this other aromatic ring. And this is tryptophan. This is what they kind of find in Turkey. It makes you kind of sleepy at Thanksgiving. And so all these different polar, non-polar, charge, bulky, small, that's all going to impact how they can interact with themselves. So here you can see two amino acids, again, going through that dehydration synthesis. This is important to note that we form what's called a peptide bond. When you join amino acids together, you're creating a peptide bond, which is why sometimes you'll hear these referred to as polypeptides, because that means you're a long chain of amino acids. It's a polypeptide. So polypeptides, once you kind of start stringing your amino acids together, once you get over about 20 amino acids strung together, they're going to start to interact with each other and they're going to fold on themselves. And this is absolutely crucial for proteins, is that the structure is going to impact its function. If proteins aren't folded properly, they cannot function. So the polypeptide chain of amino acids is not a functional protein because it's just one long chain. It has to fold. You can see in this normal protein here, it's got all these different spiral and kind of sheet planar ones, and it's all kind of interacting with itself. This is a normal functional protein versus one that's been denatured. This is what happens when we cook stuff. When you're cooking a steak or chicken or eggs or vegetables or whatever, you're breaking down those proteins within the food, and it's denaturing those proteins, making them non-functional, which is why cooking kills bacteria and things like that. It breaks down their proteins so that way they cannot be functional and they actually can't hurt us or harm us. That's why you're supposed to cook your food to a certain temperature for a certain period of time. For the one that's talking about chicken breast, right? Chicken breast, you're supposed to cook it to 165 degrees internal temperature, and it should stay there for at least 15 seconds. If you put in your thermostat or your thermometer and it gets to 165, but it immediately starts to drop, it's not enough. It needs to at least stay there for a certain period of time. Boil water advisories. Whenever you have to go through a boil water advisory because the water supply has been contaminated, you have to boil your water for a certain period of time to actually kill any pathogens. And it's because you're denaturing those proteins. And it causes the proteins to lose their shape. And once they don't have their shape, they're no longer functional. I'll also take this as a time to say there's some the raw foods movement where they say that cooking your food actually makes it harder to digest and that you're losing nutrition. That's absolutely false, completely scientifically unproven. Once we started cooking our food millennia ago, caveman figured out how to do that. It made food much safer to eat. It killed pathogens, and it already digests food for us. It goes ahead and breaks down the proteins, so our body has to do less work, and so we're actually able to be more nutritious with those foods. We spend less energy and get more calories per food, which is good. We need to do that. And so cooking your food, you absolutely should do it. So let's look at this structural stuff. I've been talking about the structure of proteins. So again, this first sequence, what we call the primary sequence, is just this chain of amino acids. And you can see each one of these R groups is a little bit different. They could be different, they could be the same. Once you have your chain kind of strung up, these amino acids are going to immediately start interacting with each other. And when they do, they're going to create a secondary structure. These are called either these spirals called an alpha helix or these kind of planar sheets called a beta sheet, right? So this is your secondary structure. It's still not really functional at this point, and this is still, you can see these kind of hydrogen bonds are causing these local interactions, these small local interactions. Right? And so all along a protein, you're going to have some regions that are spiral in these helices and some that go a little planar, and then some where the amino acids don't really interact at all. And we can start to see that here once we get into this kind of tertiary structure. So you can see the secondary part. You can see the alpha helices, you can see these flat planar sheets, and then you just have these thin ropes of amino acids where they're not interacting with each other. And so the secondary structure has been established. With the tertiary structure, you're going to start having other further apart regions interacting with each other. So primary was just a chain. Secondary, those amino acids right beside each other are going to start interacting. Tertiary, you have separate sections of a protein interacting with each other. So this is when it actually begins to fold, and that's when they become functional. Once a protein hits its tertiary shape, it can then be a functional protein. It can go do a job. It's folding in on itself. It's capable. There is one further structure, this quaternary structure. And so it would be a 4. It's a quaternary structure. This is when you have multiple proteins interacting with each other. 2 separate proteins in their tertiary structures interact together, like in hemoglobin. Hemoglobin, you actually have 4 different proteins that come together, form this quaternary structure, and are very, very capable, complex. Okay, cool. So we're going to get more into proteins. We'll use those as we go throughout most of the lectures, most of the chapters. But good thing to understand: protein structure, protein function, starting with that primary chain of amino acids, secondary alpha helices, beta sheets, tertiary into our intermolecular interactions, functional protein, quaternary, multiple proteins coming together. Next one is the DNA, RNA, nucleic acids. So this is, again, a macromolecule that is formed from monomers. We use these things called nucleotides. You see this one's called a C, this one's a G, this one's a T, this one's an A. These are each monomers that get joined together, and we're going to polymerize those to make a large macromolecule structure. DNA, if you remember, is your genetic material. That is what's passed down through our generations. It codes for everything, right? Mostly proteins. So we can go back to that. We'll learn about that in chapters like 7, 8, 9. But DNA, this sequence of nucleotides that you're seeing, C, G, T, C, G, A, this is a code for amino acids. Each one of these is in a nucleotide, right? And this sequence of nucleotides, 3 of these, is going to be one amino acid. We'll see how that translates. But this is staying in the nucleus, your basic genetic material. RNA, we're going to see, is translated. It's made from DNA for that protein synthesis process. You can see there's different structures here. DNA is double-stranded, so there's two strands of DNA, one single strand for RNA. Also, they have different nucleotides. So we're going to get into some of the specifics of that. So let's first look at a nucleotide. What is a nucleotide? A nucleotide is a structure that has three different components, kind of like amino acids have, the carbon, the carboxyl, the amino, and the R group. Nucleotides have a common structure as well. They have this central sugar. They have a phosphate group over to the side, and they have what's called a nitrogenous base. And like in amino acids, the R group was what made each nucleotide unique or each amino acid unique. For nucleotides, it's this nitrogenous base that's going to give them each their function. So the sugar and the phosphate stays the same. It's this nitrogenous base. There's some different ones. There's different options. And each one of those is going to give the nucleotide a special kind of function within that. So be familiar with these. This one you can see is deoxyribose. So this is a sugar. This is a sugar molecule. We learned about ribose sugars. We talked about that as a monosaccharide. This one is deoxy. What that means is that an oxygen has been removed. Right here on this carbon, there's usually another oxygen right here in regular ribose sugar. And so deoxyribose is missing that oxygen. So what kind of nucleotide? Is this nucleotide going to be for DNA or RNA? And if you said DNA, then you are correct. DNA is deoxyribonucleic acid. So deoxyribose. Right? Deoxyribonucleic acid, that's the type of macromolecule this is. Right? DNA, RNA are nucleic acids. RNA then is ribonucleic acid. We'll see that RNA has an oxygen right there. So it's not deoxified. Here are the 5 different nitrogenous bases. So the first 3, you find them, the first 4, rather, you see, yeah, sorry, these first 3, that was how I wanted to say it. You see in both DNA and RNA, adenine, cytosine, guanine, ACG. These are common in both DNA and RNA, both nucleic acids. This one, thymine, is only in DNA, and uracil is only in RNA. This is another way to make sure that we can distinguish between DNA or that our cells can distinguish between DNA and RNA. DNA cannot be used like RNA. If you want to actually make proteins, you have to turn DNA into RNA. And then because RNA has uracil, this is then able to become proteins. Thymine will not code for amino acids the way that uracil will. So there are 5 different types of nucleotides. And again, it's because each of these nitrogenous bases is going to give a nucleotide its functions. So similar kind of thing, when we're going to synthesize them, we go through our dehydration synthesis, we take 2 nucleotides, and we kind of join them together right where their phosphate group can meet that hydroxide. We're going to get really into the functions of DNA and RNA once we get into, like I said, the later chapters of 7, 8, and 9. We're going to really break this down. So for now, just remembering that DNA and RNA are your nucleic acids. You should learn the basic structure, review the basic structural differences between DNA. DNA is double-stranded. It has thymine and deoxyribose sugar. RNA is single-stranded. It has uracil, and it has a ribose sugar. So 3 structural differences between DNA and RNA. You should remember the general functionals differences. DNA is just storing the genetic material. RNA is actually used to make proteins. DNA stores it. DNA is turned into RNA. RNA gets translated into proteins. Okay. The individual monomer for nucleic acids is a nucleotide. So you should remember the structure of a nucleotide and the 5 nitrogenous bases. So this just really basic structural stuff at this point, nothing really functional that you need to know about DNA or RNA. The last class are lipids. Lipids are the only one that are not made of different monomers. Lipids are very different structural kind of formulas. What actually makes lipids a common macromolecule is that they are all hydrophobic. Up until this point, for the most part, these have all been hydrophilic molecules, carbs, proteins, nucleic acids. They can be in water just fine. Lipids are nonpolar molecules. They do not interact well with water. And so that's really what distinguishes them from the other classes. So here you can just see a couple of them. This is a fat molecule. This is a triglyceride. You might be familiar with that. It's triglyceride. It has this glycerol molecule and then 3 fatty acid tails. This one right here, this is the steroid hormone called testosterone. And so lipids come in a lot of different functions. They have a lot of different functions, a lot of different structures. You can see how different structurally these things are. And again, no real monomer form for them. These fatty acid tails are the same, but then this one is different, and this one doesn't really match a glycerol. So there's really no clear monomer polymerization that's happening. So we're just going to look at the different kind of classes of lipids. This first one is triglycerides. This is used for energy, right? So when you are eating something and you're getting the fat and you're getting it as an energy source, it's coming in the form of triglycerides. So these are energy rich. We use them for long-term storage. We put triglycerides, we store these in our adipose tissue. And so our body essentially breaks it down into this glycerol and these fatty acid tails, and then it can store that in adipose tissue. And then when we are exercising, or if you're in a state where you haven't been eating for a couple of hours and your body needs energy, it can start to break down this stored energy and release that to keep our blood sugar at normal levels. Steroids, another group of lipids, they're made from cholesterol. Our body can take cholesterol, modify it, and turn it into testosterone, estrogen, our androgens, lots of different things. So again, triglycerides are fats and oils. They can, I'm not going to be specific for any quizzes or tests necessarily. Well, I'm not going to say that. Well, don't hold me to that. You should still be familiar that the dehydration synthesis, you are adding this glycerol backbone to these fatty acid tails to form a triglyceride. And then hydrolysis separates the fatty acids and the glycerol. So you should be familiar with where that separation is happening. You may have heard also about fatty acids being saturated or unsaturated. So saturated fatty acids, unsaturated fatty acids, trans fats, those kind of things. You may be familiar with that. What it's referring to is how many double bonds are in those fatty acids. So if you hear about a saturated fat, what that means is that every carbon is saturated with bonds. So a carbon molecule usually can have 4 bonds around it. And so you can see 1, 2, 3, 4. All of these carbons have 4 bonds surrounding them. So they're saturated with their bonds. And what that does is that it gives it this very rigid structure. And so these things can stay solid at room temperature, kind of like butter. Butter is a very common saturated fat, lard, those animal products typically. Not always, but a lot of times. Coconut oil is also a saturated fat. Unsaturated fats, they tend to have this carbon-carbon double bond, which causes these kinks in their tails. And because of that nonlinear structure, they actually can't stay liquid at solid at room temperature. They tend to be liquid at room temperature. So things like vegetable oil, grapeseed oil, avocado oil, right? Those kind of things. And so typically unsaturated fats are healthier for you. People will push unsaturated fats over saturated fats because saturated fats tend to stay solid more. They tend to stay solid in our arteries, whereas unsaturated fats do not do that. Trans fats are when they take something that is unsaturated and they essentially force hydrogen bonds into it to make it saturated. So that way you can make something more stable for life shelf stability, preserving things, but it tends to make it unhealthy. It takes something that is healthier for you and makes it unhealthy. So you're supposed to avoid trans fats generally, or minimize them as much as you can. Where was I? Oh yeah, here we go. So saturized fats, again, saturation gives them different properties. I will say though, fat is absolutely necessary. We absolutely have to have fat, as we've already seen, for steroid production. If you need to make appropriate hormones, you need to have cholesterol for that. You have to have fats for your plasma membranes. If you don't have plasma membranes, your cells are not going to exist. Your liver needs fat to be able to process things efficiently, to be able to make certain macromolecules and other molecular structures, enzymes and things. And so without fat, you are not going to be able to be functional. And your body will limit how much fat you can actually lose. Females, women, you can usually get down to about 10% body fat before your hormones will not let you lose more. Because if you start to drop below 10% of body fat, it's going to start to impact your reproductive cycles. Whereas for men, if you start to drop below, your body actually will let you go too low. If you got below 7%, though, your liver starts to shut down. I have a friend, he's in his 60s. He has been doing yoga for probably 30 plus years, lived in Southern California. If you saw him, you would think, wow, that man is healthy. He's so fit, so skinny. Guess what? He's too skinny. Is just skin and muscle. He's been on such a low fat diet for so many decades that now his liver doesn't have enough fat and he has trouble metabolizing proteins, detoxifying his blood. So fat is absolutely essential for life. Other things that we use fats for, waxes. We don't really have waxes in our bodies, but let's say you wax your car. You're probably familiar with that, right? You get it nice and clean, you get it all nice and shiny, you throw a little wax coating on there, and then when it rains or if some mud splashes on there, it doesn't actually get into your car because that wax won't allow water to stick because wax is hydrophobic. You're seeing here an example of a honeycomb. You'll hear a lot of people tout, and it's I think mostly true, that honey really doesn't go bad because honey is so hydrophobic. There's so little water content, and bees store their honey in these wax honeycombs that does not allow water to penetrate. There's no bacterial growth. There's no water for bacteria to use to grow, and so honey can be used. A lot of people use it as like a topical antibiotic. I use it instead of Neosporin. I like to use, if it's just a minor cut or burn, something small like that, I love to just use Manuka honey. You can get it at you can get it anywhere but Walgreens or CVS or wherever. And it adds just a nice little hydrophobic barrier so that water and bacteria can't get to those places. Feels great on a burn. If you get like a cooking burn or something, it feels really nice. It's cooling, it's soothing. I love it. Yeah. Okay, keep going with lipids. We got a few more minutes. So steroids, again, steroids are lipid molecules. They are based off of cholesterol. So your liver either makes the cholesterol that you need, or if you eat animal products, eggs, fish, meat, whatever, fish is meat. I always have to remember that. You are consuming cholesterol as well, and so your liver just tends to make less. So it always tends to level out, unless of course you're eating too much cholesterol, in which case you have to monitor that. But cholesterol is important for plasma membranes. Cholesterol will embed itself into our cell membranes. That way they can resist temperature changes. So our cell membranes don't freeze up or melt when we get too hot or too cold. And like I said, they can be converted into hormones as well, our androgens, your steroid hormones that are obviously important for most secondary characteristics. Okay, I think that's most of them. Again, we're going to look at other types of lipids. We're going to look at another type called a phospholipid when we start talking about cells and our plasma membrane. But that one's not up on here yet, so we'll get that one probably in the next chapter or so. All right, if you have any questions, please let me know.